So for those of you who are new to this series, these roundtables are designed for our ABE teacher community to join from around the world. It's a chance for them to hear and learn from experts in the field and from each other about topics that are specific interests to science and biotechnology teachers. We hope that you find this series valuable, and feel free to tag us in social media if that's something you do. And if you want to share your thoughts, and I will put the tag for social media for Twitter using this tag, ABE Prog Office. So this roundtable will be recorded, as you probably just heard, and posted on our website. So if you registered for this, which you likely did if you're here now, you'll also get a transcript of the discussion and a copy of the materials that we share today. So we'll send those to you over email. We will have some time for audience questions at the end of the presentation. So I just please encourage you to put your questions in the chat box at any time during the discussion. And we will keep our eye on those. And at the end, we'll kind of gather those and go through. So your questions will get answered. So as you think of them, just throw them in the chat for us. So I am going to jump right in, and I am going to introduce our panelist. Our first panelist is Aidan Donaldson, who is currently the leader of learning for the science faculty at John Thierry Catholic College. Aidan has been a science teacher for 11 years, spending 2 years at St. Patrick's College for Girls before coming to John Thierry. Aidan believes that all students can learn and is always looking for ways to improve her pedagogy and engage students, wanting all students to achieve their very best. And of course, many of you know Aidan as an ABE Master Teacher Fellow from our 21-22 cohort. She has been a leader in ABE, and you may have seen the blog post on our website. It was titled "When Lifelong Learners Turn to Teaching," and it focused on Aidan's work and that of her other fellow, Anne-Marie Wells. I will paste the link to that in the chat here, or it's up there for you if you want to take a look if you haven't seen it before. And for our second panelist, we are delighted to have with us today another fellow from ABE Australia, Dr. Aidan Johnson. Dr. Johnson is currently the leader of pedagogy at John Thierry Catholic College, and he's been teaching in high school for 10 years, with 5 years as a science coordinator and 2 years as a leader of pedagogy. Prior to this, Aidan taught at the University of, oh boy, Wollongong. Did I say that right? Okay. While completing his PhD in forensic entomology. Aidan's always looking for opportunities to bring real science into the classroom through initiatives such as ABE. We also need to mention that Aidan was one of the very first fellows during our pilot year in 2020. He is still here, sharing his great work with us. So we'll take that as a vote of confidence, and thank you for being here with us today to you both. So we're going to begin by getting to know a little bit more about our panelists and their journey as educators. So Aidan, for those participants who don't know you, can you tell us a little bit more about your pathway into science teaching, your current school and student landscape, and the leadership role you now play with ABE Australia?
Connie, do you mind if I just share our slides?
Oh, sure.
Is it okay?
Yes, yes.
Stop the audience and go first.
Yep, it's working now. It takes a little minute.
Perfect.
Okay. Now it's just loading. Why does things take so long when you want them to be quick? Okay. My current school, as Courtney has said, is John Thierry Catholic College. I started work here at John Thierry in 2014, and my partnership with Amgen and the University of Sydney began in 2020. So my pathway to becoming a science teacher was quite a long one. I went to university, I studied my degree in science, majored in biology, did a bit of chemistry, and decided I didn't really want to be a teacher when I left university. I became a police lady for 12 years. Within my career as a police lady, I was involved in forensic science, as well as the education side of being a police officer. And then well, after I had my babies, I decided to give teaching a crack in 2011, and I've never looked back. I started at St Patrick's College for Girls at Campbelltown, and when I came to John Terry, it was a challenge, and we've worked really hard to have the science faculty that we have today that's growing in numbers and student numbers, and yeah, that's my journey. Aidan?
Okay, so similarly, I didn't set out to be a teacher. When I left school, I didn't really know what I wanted to do, but I loved science. I did a science degree, got into research and did my PhD in forensic entomology, looking at bugs in dead people. During that time, I also had the opportunity to go with my previous high school over to Kiribati, which is Pacific Island, and work with taking students over there for an immersion experience. But while I was there, a lot of the teachers were absent, and so I ended up teaching a few classes and really enjoying that. I was teaching at the university at the time as well, and had enjoyed the teaching aspect. So I sort of started to think maybe I need to get more into this teaching gig. So I did, and I think that research background has really shaped the way that I teach. And so when I became science coordinator here, as Joe alluded to, our science numbers weren't great. There wasn't, kids didn't enjoy science, they didn't want to be here. And so I was looking for ways to show students that what science really is, that it's not about reading a textbook and answering questions, that it's actually about, it's a very practical subject. And so that's when I found out about the ABE program. So we got involved with that early on. Yeah, so that was really good. And then since then, we've done other things. There's a photo of me up the top there, building a radio telescope. And we've got science extension, which is a subject where students actually get to do their own research projects. So down in the bottom right corner, there's one of my students actually getting Young Australian of the Year for the Campbelltown area for his project looking at conservation of koalas. So there's been some really great research coming through this school, which is really impressive considering, 6 years ago when we started off the journey, there was no hope of much coming out of the science faculty. So we've come a long way, and we really do put it down to this shift in pedagogy to real science in the classroom.
That's great. Thank you so much. Aidan, I'll actually continue with you. Can you tell us a little bit about why you decided to become an ABE Master Teacher Fellow and how that supported you in developing your curriculum project?
Yeah, so I think, as I sort of alluded to, my big philosophy is that science is about understanding the world. So it doesn't make sense to teach science without the real world. But often I find that we do. So just as an example, we've probably all seen this prac on the left where we pull a brick up a ramp and measure the force of it. But the kids go, okay, that was fun, but I don't know what I'm doing it for. So we've actually shifted. It's the same prac, but now we look at different shoes. So we bring sports shoes, ballet shoes, all these different shoes. And the kids see that different shoes have different purposes, and it's about the grip on the bottom of the shoe. And so it's just shifting that mindset of, okay, well, let's look at how does this apply in the real world and really do that. So I think, we were looking to improve our programs, and I said to our staff, we need programs that are relevant to the things that we're trying to teach. Obviously, we can't take topics that are way off and not in our syllabus, but they need to be relatable to the students' own worldviews and their experiences, and also needs to reflect real science. So when I heard about the ABE, I was looking, the program, the DNA component, going, how do I relate this to the kids? How do I make this real with, the limited resources that we had? And so this looked like a great opportunity. So I went and did the training just to sort of suss it out. And I wasn't sure if it would work at our school or not, to be honest. But I had a top year 10 class, so I used them as my guinea pigs, and they got so much out of it, and it ran so smoothly. And then the teachers were all quite interested in it. And so we decided that we'd train up more staff and roll it into our biology program. And did you want to talk a little bit about the success in biology?
Yeah, so what we found is the kids who have done the Amgen program through our Year 12 biology, which is for those who aren't Australian, is our final year at school. a lot of them have moved into either a medical science or a science related degree. So this year we're running it with 45 kids, I think it is, in our Year 12 cohort. And so this year marks, well, I think we've broken the 100 kids mark over the time we've been involved with ABE. So the kids find it really, really engaging. They can see the relevance of it, and they really like that hands-on experience that they get with this program. We have a lot of success with this, and the HSC our HSC, which is our high school certificate, which is the final exam that the kids do. We've been finding in our data that it's the highest that this is where the kids are achieving their best in the final exam. So we've had a lot of success with this program.
That's amazing. And I'm going to have you continue a little bit, because you both spoke about how equity and access is sort of a priority for you in your teaching. It's also a priority of the ABE program and the Amgen Foundation. Do you still have goals that you are looking at for increasing access? Do you feel like now that you've sort of implemented the ABE program and you've done the MTF project, are there things now that you're starting to zone in on that you would like to target?
Yeah, so I think this slide sort of sums up where we've been coming from and where we're still working towards. So we're looking, rather than trying to develop students' knowledge as such, we're looking to move students to that application of knowledge. And we've found, the evidence has shown us that the students are learning science by doing the science, and working as scientists would work. That is, so working scientifically is how you actually acquire scientific knowledge, rather than reading and teaching the knowledge. And so that shift in pedagogy has been really strong. We're still working on trying to get the students to access exam questions, I suppose. The literacy of our students is quite low. And so the practical component of it is there, and they're developing the knowledge, but then how do they transcribe that onto paper? Can I talk a bit more about that? Yeah,
I think it's that explicit teaching. So teachers are forever updating programs and things like that, of course, but we're actually backward mapping a lot of our programs and having a look and see where aren't we explicitly teaching these skills to students. And if we go back and explicitly teach these skills to students and get them to practice them, we find that they're becoming more confident in themselves, more confident in their own ability to apply science, and then they enjoy science because they're having success. So it's I suppose that's what we're working towards at the moment.
Perfect. Thank you so much to both of you for sharing those stories and your experiences. We know that you have some wonderful show and tell examples from your projects that you're going to share with us today. So we always like to be able to share these practical examples and resources for the participants. So for those in our audience, we'll have some time for Q&A at the end. So if you have any questions you'd like to ask our panelists as they present, please write them in the chat and we'll be sure to ask them at the end. So I want to invite Joanne, if you want to share your screen and walk us through your project and how teachers might adapt or use it to enhance their own science classroom and lessons. And I will put a link, no problem. I'll put a link to your project in the chat for those that would like to access it.
Okay, so my project for my curriculum project was I suppose I created this curriculum resource as a need, because the topic scientific regulation can be a little bit dry for students, and we were looking. I was looking at a way to engage the students, but to also enhance their literacy skills as well as their research skills. So the students came out of the project. The students have actually tested this project and they enjoyed it. They said they enjoyed it, and they needed to the program or the project explicitly explained, the aim was to explicitly explain to teach students how to explain scientifically. So they, by the end of their goal, by the end of the project, was to be able to explain the need for scientific regulation, explain and assess the ethical issues. Then they need to do a compare and contrast. And then at the end of their research, their goal was to evaluate the effectiveness of this international regulation for scientific research. So it was a very structured project, and it also instructed students how to work in a team, I suppose, how to work as a group, and how it's important to include all group members to be involved in this research, otherwise you weren't going to get much done. So I can't remember, no.
I'll just butt in on that.
Yeah.
So this was actually, Joe taught my class this, so I actually got to observe this lesson in action, which was really good. But something I noticed as their teacher is they weren't great at group work, and when Joe came in and sort of explained what they had to do, at first they just went off and worked in their silos, and they were going, so this is really hard, I don't know how to answer this question because I don't know the answer to that one. And I'm like, that's why you're working in a group, because someone else is answering that question and you're meant to be discussing your answers and talking through what you're doing. And like, oh, so, you know, that group work idea for some of them, particularly I think, you know, we came back from remote learning, so they'd had 2 years where they hadn't really been in a classroom, let alone doing group work, and then suddenly they were doing this large group project.
So yeah, so it was a very, the feedback I got from the kids was really good. The feedback I got from Aidan was good. So it was sort of, the kids really enjoyed doing it, and the fact that they got to engage with each other as well as the content, which sometimes I think could be just delivered to them as a lecture style, was quite good. It was good. It was good for them. It was great for me to get that good feedback. So yeah, so that's my.
And again, in terms of their final exam results, that component, they went quite well in the final exams. So it worked.
Yeah, it worked. That's good.
That's perfect. So Aidan, I'll turn to you next. I didn't know if you want to share some example from your project, which was Stage 6 Science, Investigating Science. And I will also paste this link in the chat for you guys to explore.
Okay, so my project was, we've got a subject here in Australia called Investigating Science, which really is, it really lends itself to my passion. It is the skill set of doing science rather than the content of science. And so it talks about the role that technology has on science and the role that science has on technology, and that these two things are interrelated. And so when I started the fellowship, I was sort of thinking about this module going, you know, there's so much scope here, but where do I go with it? Because it is a very open-ended sort of topic. And so I started thinking about DNA and the developments of CRISPR. So I started looking at the journey to get to CRISPR, and I realised, well, we started with X-ray crystallography, which allowed us to understand the structure of DNA. So the technology informed the science. But then that science, knowing the structure of DNA, allowed us to do the Human Genome Project, and through that developed some new technologies. Those technologies then allowed us to have a better understanding of genetic sequences of disease, so new scientific knowledge, which has led to this new technology of CRISPR. So I thought that was a really cool way to show that it's not that technology leads to more science or that science leads to technology, but actually they both keep building on each other. So that was sort of my focus, obviously biting off way more than I could chew at the start. So I ended up focusing just on that first step, X-ray crystallography to structure of DNA. I'm still developing the later ones because I want the whole sequence, but for the project, I focused just on that first step. And what I've developed is the scrollable, after CRISPR comes treatment of genetic disease, obviously, is a scrollable for lab exchange. Now, this is still a work in progress, and I'm hoping we can finalise it in the near future. But the bones of it are there, the structure's there. We just need to sort of work with the animators to sort of finalise it all. But the idea is that students can scroll through this and see that journey that I just sort of unpacked. So looking at, what is X-ray crystallography, understand the technology a little bit, and then understanding, how it was developed, looking at X-rays, and then that they can be used to find structures, and then looking at the crystal patterns, and then just understand a bit of the science behind that technology. So even though that's the first piece of technology we're looking at, there's still science that informs that. And then looking at how that actually works. So unpacking the science there, diffraction patterns, and then looking at how that can be used to predict structures and shapes. That video is not working, but that's just a video of some diffraction patterns actually in action. And then the idea that different structures will produce different patterns. And so then we've just got a couple of examples of zinc sulfide, and there's the pattern, and that's the structure. And then lysosome, structure, pattern and structure. So just giving them a lot of examples so that they understand, because I think when we show that photo 51 and we go, oh, from that they worked out the structure, and the kids just look and go, sure, I don't know how that happened. So showing that every structure has a different pattern to start with, and then, they start to understand that, okay, well, if every structure's got a pattern, then this unique structure also has a unique pattern. And so just talking about that discovery, bringing Rosalind Franklin in, bringing her front and centre of the discovery, because without her photo, we wouldn't have found much. And then just unpacking a little bit about that photo 51, and then the role that Watson and Crick played in sort of modelling that out, taking that information. And then, so the idea is, as they work through this, we're really, I mentioned that, we're trying to develop students' ability to actually answer questions with this knowledge. So they develop the knowledge, but want them to understand that scientific idea. So what is it? And then what's the contribution that scientific idea has had to technology? What's the new technology that came from that? Then make a judgment about the importance of the scientific idea on the development of that technology. And then look at the contribution that has had to science, and then what science has come out of that technology, and then make a judgment about the importance of the technology. So it's really guiding students through how to really analyse and assess and make a judgment about what is happening in science. So that was my project in a nutshell. And again, as I said, it is still a work in progress 3 years later. But the structure's there, and I still use that more in a PowerPoint form, but one day it will be a scrollable, I promise.
Thank you both so much for sharing these just rich examples and ideas. And I'd like to remind everyone that you can access these projects as well as any others that were developed by Master Teacher Fellows at our website, and I will post that link in the chat here. But they are all housed there, and there's quite a few that are just amazing, but these two are just really stood out to us as something that could be adapted to the classroom with other ABE teachers. So we hope you take advantage of this. So I want to take some questions, and we don't have a too large of a group. So I'll open it up if you'd like to unmic and ask a question, or if you feel more comfortable, you can also type your question into the chat and we can share that with our presenters. But I'll give everyone a moment and see if we have any questions. Well, I will kick us off, and then maybe that'll warm up everyone. I'm curious if there was like a common challenge or something that you experienced that surprised you when it came to making DNA concepts hands-on with kids. Was there something that you found was unexpected, that maybe a misconception of theirs, or something that presented a challenge that you weren't exactly prepared for?
I think one of the biggest challenges with DNA is just getting kids' heads around what it actually is. We talk about it all the time, like everyone knows what it is, but they get confused, you know, where are the genes, where are the chromosomes, where's the DNA, you know, is there different DNA in each chromosome, or is the chromosomes got the same DNA but different bits? And so just unpacking that, the general structure, I think that's where, you know, those general misconceptions come, even though we do go through the structure and, you know, how it is used throughout the different levels up to cell level, they still sort of get confused as to, okay, which bit goes where.
And I think they also, like, because we're actually doing the ABE right now, Teresa and I are in the middle of our biotech experience right now. So our transformations are happening as we speak up in the lab. And what the kids it blows their minds is that protein synthesis is the same in a bacteria as it is with us. And they just go, what? I'm not the same as the bacteria. Yes, you are. And that's that common thread. And it just blows their mind. And because I said to them, we're looking at the plasmid and how is this new protein going to be made? And they're like, we don't know. Oh, come on, guys, think about it. And what about transcription and translation? That doesn't happen in a bacteria, that happens in us. Well, yeah. So it's that. It blew their minds. It was like, oh my God, that blew my mind. And they came up, a couple of them came up to me after class and said to me, Miss, that really opened our minds to how scientists think. You know, if this can happen in us, is this happening in another organism? And what can we do with it? And at the end of the day, that's how we want them thinking, you know. And I thought, thank God the pennies dropped. Yeah.
Well, and as they walked by a bug or some kind of organism, their brain went to, oh my gosh, that's happening in them right now.
Yeah, that's right. Right now.
Yes.
Yeah.
To see it in their own environment is so important.
Yeah.
I'm curious what barriers you think may exist for classroom teachers when it comes to using cutting edge technology in the classroom, and if you have any advice for those teachers in addressing these barriers.
I think at the moment, our attendance rate is our biggest barrier. At the start of the experience, I had half of my class there because they had COVID. Half of them had COVID. And I think there's no way for us to get around that, obviously. But I think the other barrier would be the area that we're in a low socioeconomic area. And the kids, some of them, the culture is changing. They don't place education as high as what they should. So you say to them, this is really important for you to come to school. It's, we really need you, especially over the next 4 weeks, to be here every single day. And then I get an email the next day, Miss, I'm away for 6 weeks because I'm going to visit family in the Philippines.
Interesting. It's so different than the US structure. I'm interested just in that.
And she said to me, so Miss, what are you going to do about my assessment task? And I went, mate, what am I going to do? But this mindset is changing at the school. It's very much a cultural thing, and I think that cultural change is changing. The teachers at the school, the exec, are really fighting to make that change, and that change is very slowly happening. I can see it in my year 11 class, my year 11 biology class. I've got a massive cohort this year, and already we're 5 weeks in and they're already in a routine past paper questions on a Tuesday, and every single one of them does it. The attendance rates are really good. So it's taken us maybe 5 or 6 years to get that culture of learning moving, but it has improved. So great. And it's that culture of learning that you've got to harness.
I think too, just with the question, like coming from the different angles, so the students are the challenge, but then for other teachers, I think, you know, there's their confidence in their own ability to use this technology and what, you know, I don't understand it fully. How can I teach, you know, second year university stuff in a high school? You know, will my students be up to it? Will I be up to it? So I think my advice to those teachers is just have a go. I think when I started off, you know, I did it in my second year at this school, and this was much before the culture had shifted, and this was a situation where we had enough beakers for one class, not 6, because the kids would just smash them, and here I was borrowing very expensive equipment from a university, and I was very nervous. But, you know, you explain to the students, and obviously I picked the top class, the kids that are most engaged to experiment with. But now we're at a point where, you know, we experimented and it was a success, and other teachers, and I had kids from other classes go, can we join your class? I was like, no. But they all wanted to learn science, and they all wanted to. And so, you know, and since then we've now bought some gel electrophoresis tanks, and so we do some micropipettes, so we're using it. And again, when we bought those teachers, like, oh, I can't do that with my class. I'm like, yes, you can. They will be okay. You explain to them how it works, explain to them how expensive it is, and they will do it. And, you know, yes, we get a pipette break here or there, but it's worth it.
Yeah, we get them. And last year we had, with our gel tanks and our micro pipettes, I rewrote the program to be around a case study for DNA profiling, and teachers, like as Aidan said, can't do that. There's no way I could do that. And I'm like, yes, you can. Just go in there and have a go. It's going to be okay. If you stuff up, who cares? That's how you learn. And once they realise that this actually works, and the kids are engaged in it because they're like, oh, there's a story behind this, and it's a profit. And look, science has helped these people. And you know, the teachers are engaged. When the teachers are engaged, the students are engaged. So
it's that role of both doing science, but then also the stories that go with it.
Yeah, that goes with it.
Really gets the kids in.
We had a couple of questions. One was if your school provides technology devices for students.
So we're a one-to-one school with the technology, but the students bring their own devices. For being low socioeconomic, obviously there's some families that can't afford it, so we do have a loan system, so they can loan it out for a daily loan if they need to, or over the term. So it's sort of worked out on a student-by-student basis. But generally we generally use Chromebooks because they're a lot cheaper. So that's sort of how we make it accessible. But we do have loans if needed. And
then the kids tend to forget to charge the devices because, you know, they get distracted, or, you know, home's not great. We've actually got a charging pod at school. It's like a big filing cabinet. The kids put it in a secure locker and then they can charge it. So
perfect.
Yeah.
Okay. The other one, not so much a question, but I found this. I actually found an aha moment in this, that we eat DNA every day, which is very true, very meta thinking. And so I'm curious, have you done any DNA extraction with fruit and then sort of landed that bomb on them that they're now eating DNA? Yeah,
we do the DNA extraction. We use peas and strawberries. Strawberries are really good. And kids have gone, but I eat more strawberries.
Great. It's always fun to sort of rock their world a little bit when they're so hard-shelled sometimes.
Love it. It's so good.
I turned off my mic to laugh out loud at that one, because when I was a teacher, I taught biology, and we did that DNA extraction from strawberries, like you guys said. And then they're like, oh no, is it safe to eat strawberries? And then it's in everything. So such good aha moments.
Yes. Salvatore had a good point here about the importance of reflecting in the classroom about the relationship between science and technology. So students sometimes have the wrong ideas about technology. They think that those things are used mainly in electronics and energy. But he mentioned today, he focused his lesson about the role of electronic microscopes and technology in the discovery of viruses. And I think that's so true with students today, especially. When you say technology, they think of their phone or their iPad or, you know, those sorts of things, and not that these mechanisms are used in daily discoveries in medicine and biotech. Do you bring any of their technology into the classroom, any of their technology applications into the classroom?
Yeah, I mean, the phones are a great scientific device these days. You know, there's all sorts of Google Lab and all sorts of things you can do with measuring pitch, volume.
Make the 3D image from the prism.
Yeah, 3D imaging.
Yeah, there's the 3D imaging from, I don't know if any of you guys have done it. You get a bit of perspex and you make like a pyramid without a top on it, and then you put it upside down on your phone, and there's an app, and as it shines the light through, it makes makes a hologram.
Yes. Oh, perfect.
Yeah.
We do have another question. Ashley is curious if and how you're using Lab Exchange in combination with the ABE labs.
Yeah, so I'm a bit of a control freak. So when I run ABE, the weeks beforehand, I map out. I have a calendar for my Year 12 kids, and I use Lab Exchange. They do their Lab Exchange before they come into the lab. So they have some lessons to do. But Teresa and I have just incorporated it into my biology class, my Year 11 biology class. Teresa just showed me how to set up a lesson, and we're also using it in conjunction with, I will speak to the kids obviously at the start of the period, and then they self-paced and they work through some lessons on Lab Exchange. So we've found Lab Exchange really, really helpful. We really like Lab Exchange, and the kids love it too. The feedback I've gotten from the kids is that they like how it's interactive, and they love how they can work at their own pace and they get immediate feedback. So that's what they really like about Lab Exchange.
So that flipped learning idea, which is funny because we've been trying for years to get a bit of a flipped learning culture, and it just hasn't happened. And then suddenly, the kids are like, "Oh, well, this is good.
This is
Let's
fun.
do this."
I like this.
Like, yes, please.
Perfect. I'm curious what advice you have for teachers who are new to ABE.
Map it out. Map it out. So if you've got the equipment, like we were lucky enough, Eugenia lets us have it for 4 weeks. So I map out, I've got my lessons planned right down to almost the minute, but that's me, a control freak. And I have it, I know exactly what I'm doing, and when I'm doing it, it's really detailed. That was my first ABE. Now I'm an old hat at it, not old, I've done it many times. I know what I'm doing and I understand the flow of it, but if you're doing it for the first time, I would really strongly suggest mapping it out, like on a Google sheet or an Excel spreadsheet. Have the days of the week when your Year 12 have your class, and then work out that those incubation times are the big ones. So if you're going to have your incubation, if you're going to do it over a few weeks, it's the incubation times that we try to map out first. We have a look, those really long 2-hour ones up for the PCR, when the PCR runs. Just make sure that you give the kids time. You give the kids time to learn how to micropipette. You give the kids time to be able to understand how to load those samples into those gels. Because if they get that micropipetting wrong, the whole thing doesn't work. It doesn't, you run out of reagents, you run out of everything, and the kids stand there, why didn't it work, miss? Well, it's your micropipetting skills. So.
But if it doesn't work, that's okay.
It's okay. That's what I say to them. Look, if it doesn't work, you've got more to talk about in your report. And And, you know, it's a wide, and then we do, last year, we had a couple of kids who, beautiful children, absolutely wonderful children, but they didn't have 100% concentration. So we only had 2 groups out of the 6 that were actually successful. And they were a bit down. And I said, but what did you learn from this? Let's sit down, let's have a chat. What did you learn? Let's work out how you could have done this better. And then I think they actually, upon reflecting upon their skills and their science, they went, oh, this is because of this and this, and they actually came out understanding it better because they made a mistake.
That's amazing. It's true,
Yeah,
though. Yeah.
that is true. Yeah.
Sometimes not being easy makes it the best lesson when they have to go back through and think. And any other questions from our audience? Anything else that you two would like to add?
We did apply for the Amgen grant, so we talked a little bit about that. So we were successful in getting the Amgen grant. So that's sort of, we're using that as our next step, I suppose. So we're looking to, actually I've got a video, how do I project to that?
If you go, you know, where we go up here with the 3 dots.
Yep, okay. Sorry, I'll just connect this quickly.
Sorry, that one. And then fishbowl.
So basically what we're looking at is that modelling of DNA, the process of discovery. So it'll sort of tie in nicely as the next step in my fellowship project. And the idea is I've been doing this prac, which I'll just show you now and I'll talk you through. So basically using a laser just to model the diffraction patterns. And so there's the equipment that we need. So it's all pretty straightforward equipment. You make your coil, your DNA strand, just out of some nichrome wire. And I make it look really easy in this video, but it's a bit of trial and error. So the idea is that that's now your helix shape. And then we're using the laser to generate our light source. And so the idea is shining the laser through that helix and you get a nice diffraction pattern. You can see that X pattern there. So showing this to the students, they kind of can start to see, and then I start to play with the spring, stretching it out and compressing it, and they can see that the size of that X then changes. And so I was explaining to them that this is how, you know, they understood the X pattern meant that it was a helix, and then this angle and size of that X actually reflected the structure of DNA. So that's a really good prac, but as I did mention, it's quite fiddly. And it doesn't always work, and it's a lot of trial and error. And as you can imagine, it works perfectly when you don't have kids in front of you, and then you try to do it in class, and you can just bear with me, kids. Hang on. So part of what we're developing with the Amgen grant is actually we're going to get a laser table and set the laser up, so it's all set up properly and stably rather than trying to line things up perfectly, so that this prac will actually be doable by any teacher, hopefully. If it works the way we're hoping it will, anyone will be able to just set it up quickly and easily, because it is a very effective demonstration, but as I said, it can be quite fiddly.
Yeah, that's really neat to see. Yeah, that's really neat to see. I got to see your application on paper, but it's neat to see what it looks like in application in real life. So I'm curious, we did have one more question here, and it was, what was a highlight from your time as a master teacher fellow?
For me, I think it was having those discussions with people all around the world, and just understanding how the different education systems work for a start, you know, and hearing what sort of science is taught in different parts of the world. You sort of go, oh wow, you teach that in high school, we don't touch on that, or, you know, so just hearing the way different systems work and sort of having that international collaboration is not something you get exposed to through any other program I've been involved with. You know, I try and get involved with as much PD and things as I can, but yeah, the international experience is very different. So that was my highlight.
Yeah, I think we spoke last night, like yesterday afternoon, in a meeting about our involvement with ABE, and we that's what I said. It was that involvement with international teachers. You know, speaking to them about, you know, what are their kids like? Their kids are the same as ours, you know, like they were flat, and they, you know, at different times of year, you know, and having other faculties come up and go, how do you get involved in something like that? You know, like it, and I suppose Aidan and I have sort of, other people want to come and not join, not ABE, obviously, PE faculty, but to be involved in something like that, like this, but other faculties, I think that's a real highlight. It inspires people to want to do stuff that's sort of outside the school, outside the school, which is, yeah, and it benefits the school too, you know, which is great. So.
Well, you guys have been great proponents of, because we are now in our third Master Teacher Fellow from your school, which is so neat to see the scalability and the sustainability that you guys are implementing within your school and ABE Australia. And just like people have said in the chat, the progression of your work from your project to your mini grant and the application of, you know, the materials, it's really exciting to see how you guys have taken this and ran with it the last 3 years.
Yeah, we have run.
It's my mini PhD.
Yeah. Well, if we don't have any other questions, I just wanted to say a few things. I want to say a final thank you to our panelists, Joanne Donaldson and Aidan Johnson. We hope that this roundtable gives you some ideas for your own classroom, and you are also more than welcome to visit us at the ABE website, where you can find their projects housed there. Next month, as a heads up, on the 28th of March, we'll be hosting our roundtable on peer learning models for ABE teachers, and we are going to be joined by teachers and program site staff from ABE Italy and ABE Turkey. They are going to share some of the really wonderful examples they have of how teachers are learning from other teachers within their ABE program. They're going to talk about how their teachers work together in communities of practice, how they observe and share feedback with one another, and also discuss with us how they develop as leaders within the ABE program. So please do join us for that. We'll be sharing a registration link for that very soon. As we know, the ABE teacher community is really what makes this program thrive and grow. So we want to thank you all for being here, for everything you do for your students. And it's just so inspiring to see the excitement come through from the teachers, because you know that that gets taken into the classroom, and that's really what makes those wheels spin. So I would also like to say a huge thanks to the Amgen Foundation for supporting the Amgen Biotech experience. I hope you guys have a great rest of your day wherever you are in the world, and I thank you for joining this ABE Teacher Roundtable.
Thank you.
Thank you
So much.